Welcome back, everyone. Again, one of the series of our XMAS tutorials. What we want to do today is after we got into some basics, or so now we know how to handle structure, map crosslinks, get some basic statistics and explore the data, import, export, molecular modeling, and so on. We want to fully stress our software um, with gigantic protein-wide data sets on a normal laptop, as the one I'm using now. So we want to bring this software to full capacity, but we also want to explore the amount of data that is out there, and we just start to scratch the surface. So how we can combine now powerful tools for structural predictions and cross-linking data that we have already. So to do this, I will give you two examples in this first video. We will look first into this Xlink DB. This is 4.0 version. This is a wonderful resource from the Bruce, curated by the Bruce Lab. What you can do here is, for example, a species view, and you have several species here with different data sets that are all publicly available, but here they are completely annotated. You can look them through so just molecular networks, but we can also generate a XLMS interaction table for Homo sapiens, for example. This may take a while, so we can download the data table, and these steps may take a while because it's 18,000 different interactions. I can show you how it looks like. So it's here. Oh, here we go. This is XNX output human. So you will have it here. You can see it's a quite gigantic amount of data. So we are talking about 10,000. Oh, here I applied some filters. Here we go, 19,400 lines. So from these, we can extract some of the proteins we might be interested in. So you have a lot of histones, we might expect this. Let's say we want to look at cytoskeleton, so like tubulins. So what we can do is, for example, from this big table, we can extract all proteins related to the cytoskeleton, let's say actin or tubulins. And for example, we just by symbol copy pasting this accession number, we have a list of unique proteins. In this case, I selected 59 that are involved in the cytoskeleton functions. And by simply adding this AlphaFold fetch command, which is internal command of ChimeraX, they just added to fully improve the usage of this AlphaFold predicted proteins. So we just copy paste this list of command. It's as simple as that. And we copy paste in the command line of ChimeraX. And wait a bit while it's downloading these 59 structures. Takes a few seconds. We have them all here. You see them in the model panel. Now it may look a bit messy. What we can do is simply type the command tile, and boom, it will order them and tile them in different ways. And then what we only have to do now is to import the table. Just take care of the naming on the columns, on the headings, but it just needs peptides and linker position of the one off position of peptide A and peptide and linker position of peptide B. Oops. So now the only thing we have to do is to import file and map the crosslinks against all our structures. And boom, we have 376 pseudo bonds, and we have them all here. It's quite impressive. And from these, you can then start to look at specific proteins. Of course, this looks a bit messy, but just to show you that in a few seconds, we can get from publicly available data sets and map to structures that are predicted with high confidence. And we have no issue in handling a huge amount of structures and data on normal, a very normal laptop. And in the second part of this tutorial, I actually want to show you another application. So for example, for this case, I pick it up publication. So this is a publication from Yuri Rab Silver Lab, identification of protein-protein interaction by cross-linking mass spectrometry. This is a beautiful paper on true FDR control that I recommend you to read, but it's very maybe very technical. What I was interested in is how they use this dataset to find uncharacterized proteins, this YAC-L. They use E. coli full lysates, and they also generate quite huge data sets because they fractionate protein complexes using size exclusion, so in native conditions, and then also peptides are highly fractionated. So we want to test this if we can confirm, for example, this hypothesis. Well, let's say the findings they have on this complex, the binding to RNA polymerase. So if you scroll down in the sections, the beauty of the mass spectrometry community is that we always have the raw data available. So just dig into this data availability section until we find proteome exchange or JPOST. In this case, we can use this PRIDE repository. It looks like this. So this is a project. It can be difficult to really find the files you really need. But in the FTP sections, we are going to grab this E. coli DSSO unique proteins extra modification, and also this E. coli VS3 file. It's downloading. They are quite heavy, like 20 something. So I can show you how they look like. And this is the E. coli DSSO. It's 38,000 lines. So quite a huge data sets. The BS3, I think it's more or less the same. Oh, it's only 29,000 lines. So what we can do now is just look where did they what was the PDB used for this figure. Here you can find it in the caption. PDB is 6C6U. So 6U. Here we go. And then in this case, we only need to open it. So we can just say open. And we open the RNA polymerase. And then this NUSG and YAC-L proteins of E. coli. Again, we can fetch the alpha folds. So we just need to type alpha from fetch and the name and the UniProt ID. We do the same for YAC-L. Here we go. Okay, now we're good. We can, I will show you later. So the why I downloaded the AlphaFold model for NUSG. This is also quite an important point because many times we have missing parts in PDB structures. That is not a negative aspects. It's just that there are limitations in structural determination approaches. So some part of the structures might just not be there or not be resolved. And this is one of the case. So if we, for example, match we go to tools, for example, let's do the alignment. So if we do structure analysis, a matchmaker, and we want to align this with the structure to match on the RNA polymerase, this should be already one of the subunits. So show pairwise alignment and it's apply. What you will see is here, look at the alignment. It aligns almost perfectly, but there is a big part of the structure that is actually missing. So almost all the N terminus aligns, but the C terminus is not there. And we will see whether it makes sense to have the full structure or not once we start to map the crosslinks. So now we import our crosslinking table from this huge experiment. So remember that now we have 40, we are almost trying to match 80,000 different lines where we need to align every single peptide sequence. So I will hit the map crosslink button, but it will take a while. Here we go. So it was done in just our past 2 minutes and something. So you can see a lot of intralinks in the RNA polymerase were formed, and here we see 5 interlinks between YAC, L, and RNA polymerase. So what I wanted to show you here is that if we just move away this from the subunits of the RNA polymerase, like this NUSG that we got from AlphaFolds, you will see that actually a lot of the data comes from these C-terminal parts that we actually couldn't see, that was not actually present in the full model, but that really nicely comes together when it goes into the full RNA polymerase structure in the whole complex, let's say. So if we don't consider that we may have data for full proteins, but we don't have structure for full proteins. But here, AlphaFold can really help in getting very new insights. Well, with these crosslinks, what you can do, it's as we did before, plot, for example. Well, we can get the chosen name because the names of this file are incredibly long. So let's call it BS3 and DSSO. And we can again plot the overlap, for example, between these two data sets, let's say only from the crosslinks that we extracted and then match for these three structures that we have open in our session. And the distances, for example, of course, now we have very long ones. You will see that if we approach back the molecules, of course, these distances will be much slower. So let's put this back where it was, and then you will see that everything will come together again. Still quite some long crosslinks, but that's understandable. RNA polymerase is very flexible, so you can see where these very long ones are actually more or less located. Yeah, these two very long ones. Okay, thank you for following this series of tutorials. I hope you liked it. If you have any question or doubts, you can contact us through GitHub, the GitHub repository, or directly by email. If we have new cool developments, for sure we are thinking about many different stuff, so we'll update with new tutorials or new announcements on the XMAS webpage. Thank you for following. Bye.